welcome to Edukemy's YouTube channel. Myself Karuna Mishra, and we will be continuing our previous year question series. We have already taken 2 questions from Science and Technology. Both of the questions were from Information and Communication Technology. Today, I have taken 2 questions which are also synonymous to the Nobel Prizes which were given in the year 2020. Now, whenever you read Nobel Prizes, you do not need to remember the name of the Nobel laureates. If you know it, it is very good. But for UPSC, you have to know the technology or the area in which they have studied and the basic science behind it. So today I will be taking the previous year question of the year 2020, which were also synonymous with the Nobel Prize in medicine and in the field of chemistry. First question is regarding the hepatitis virus. It says that which of the following statement is not correct. So the first statement is that hepatitis virus is transmitted like HIV virus. Alright. The second statement says that hepatitis B, unlike hepatitis C, does not have a vaccine. So this means that hepatitis C has a vaccine, whereas hepatitis B does not have a vaccine. Even if you do not know about the basics of hepatitis, I think you remember if you got the vaccine when you were children or not, when you were kids, when you were of the younger age, especially there were drives in the schools also where the hepatitis vaccines were given. The question is, what was it, B or was it C? The third statement is, globally the number of people infected with hepatitis B and C are several times more than those with HIV. So this is more technical, that the combined number is more or not. The fourth one says is, some of those infected with hepatitis B and C do not show the symptoms for many years. Now, in order to answer this question, we need to know about this virus. So let us start. First of all, I would like to tell you that the Nobel Prize in Physiology or Medicine 2020 was given for the discovery of hepatitis C virus. So now you know the rationale why UPSC gave this question. These are the 3 Nobel laureates. Again, do I need to remember them? No. So let me just understand that since hepatitis C virus was discovered, or it was not discovered recently, but it was awarded. So let me understand what this virus is. So hepatitis is an inflammatory disease which causes damage to liver. Hepatitis viruses are the most common cause of hepatitis. That means the disease hepatitis is not just caused by the hepatitis virus, but can be caused, alright, the most common cause is the virus. The other causes are alcohol, drugs, and autoimmune diseases. So apart from the virus, the other causes for the hepatitis diseases, hepatitis virus, alcohol, drugs, and autoimmune diseases or autoimmune condition. So one, I understood that it causes damage to liver. Second, I understood that apart from the virus, these are the other factors. Now, something else that I need to know about hepatitis is that it is a blood-borne virus. When I say blood-borne virus, that is whenever the infected blood is transfused into an individual, it causes the hepatitis C disease. Now, I am more concerned about hepatitis C disease or virus because it is hepatitis C which received the recognition and Nobel Prize. Now, this transfusion of blood as well as blood products. Now, what are your blood products? Your FFPs, your platelets, your plasma, all of these are called my blood products. Apart from that, sexual transmission is also a cause, but is not the most common cause. In HIV, it is one of the most common cause. So that makes hepatitis C different from HIV. About the number of people, it says that around 71 million people, of which 6 to 11 million are in India, which are suffering from hepatitis C virus. Also very important is that it is a major cause of liver cancer, and no vaccine is available for hepatitis C. So hepatitis C does not have any vaccine available for it. So we got all this information about hepatitis C virus. So I can see that what actually happens when the body is infected with the hepatitis C, hepatitis A, B and C, these two are blood transmitted. Hepatitis A is not just blood transmitted but can be transmitted through contaminated food or water also. Now, my liver, my healthy liver and my chronic hepatitis then leads to cirrhosis and then finally carcinoma, which is the cancerous stage. Cancerous stage is achieved in the liver because of the infection from this virus. So I can conclude that of these hepatitis viruses, which is hepatitis A, B, D and E, we have already discussed about C. I have vaccine availability for all of them, but I do not have vaccine availability for hepatitis C. Now about hepatitis A, as I said, it can be spread through contaminated water and food, and also sexually it can be transmitted. Coming to hepatitis B, which also received Nobel Prize in the year 1976, is due to the exposure to infected blood, semen and other bodily fluid from infected mother to infants at the time of birth. Also transfusion of hepatitis B contaminated blood and blood products. So this is, and most common, one of the most common is the drug use. The drug use, when you use same needles, among different people, and if one of them is hepatitis B virus infected, it creates the spread of virus among the people. So drug also is an important factor here. Hepatitis D virus is similar to hepatitis B virus, and hepatitis E virus is due to contaminated food and water. I have vaccine availability for all of them. So going back to the question, so I can say that it is statement B which is not correct or is the right answer, because hepatitis B has a vaccine but hepatitis C does not have a vaccine. So this was about hepatitis virus. Now coming to the next question, and another very important technology on which different questions have been asked repeatedly. My next question is from CAS9 Protein. It is related to Crispr technique. A lot of questions in the past have been asked over Crispr. It is a perennial topic, will always remain important. Now CAS9 protein is one of the factors which helps in Crispr technology to work. So let us first see the options. The option is, it is a molecular scissor used in targeted gene editing. Second is, it is a biosensor which is used in the accurate detection of pathogens in the patient. C is, it is a gene that makes plants pest resistance, or it increases the pest resistance in the plants or makes them pest resistant. D is, it is a herbicidal substance synthesized in generally modified crop. So I know one thing, these two are related to BT. Agreed? So whenever I read about BT, Bacillus thuringiensis, BT crops, BT cotton, I find these things. This is BT cotton, the latest. Tell me in the comments, do you know that BT cotton has a new version now, which is now herbicidal substance synthesized? Is it right, or there is something more to add to it? So this is related to my BT. Coming to the biosensors used in accurate detection, this is more on the lines of finding the disease, whereas CAS9 is actually a molecular scissor. Now let me understand what is CRISPR technology, and then I will come back, or CRISPR technique, and then I will come back to this question. Now, my 2020 Nobel Prize in Chemistry was given to CRISPR, and this is what CRISPR technique is about. So I have palindromic sequence. What is palindromic sequence? It is, if you read it from the starting or from the back side, it is same. So that is a palindrome. I have found this area. CRISPR basically stands for clustered, regularly interspaced, short palindromic repeats. So these are the palindromic repeats. what am I doing? I am basically using a guided RNA. Now remember this technique, because if you are trying to understand the mRNA vaccine, there also I am using the guided RNAs. So I am using the guide RNA. CAS9 here acts like a scissor, which is an enzyme. It cuts the DNA from the target area, and then another enzyme here will repair the cut DNA. The DNA will rather either repair itself, or I will add the desired genetic material or desired section of DNA in this area, and this is how CRISPR technology works. So what is my role of scissor here? The role of scissor here is basically snipping. It is the snipping tool over here. Here the cell will try to repair the cut DNA, or I can also add the desired DNA here. Alright, so this is how my CRISPR technique works. Where am I using my CRISPR technique? I use it for plant breeding. I use it in different type of cancer therapies, and I also use it for curing the genetic or hereditary diseases. So I am using my CRISPR technique for all these areas. So these were the two questions which we discussed, which basically come under the biotechnology part, but since the Nobel Prizes were also given on this was very important. So I hope you got an idea. This video helped you. If you want more such videos, let us know. Thank you so much. All the best for your prelims.